Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask the speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. Thank you for joining us today. It is now my pleasure to introduce our speaker, Olivier Berteau from METTLER TOLEDO Process Analytics.
Thank you. Thank you for having me here today. So, hello again, and welcome to this 15 minutes METTLER TOLEDO Process Analytics webinar on dissolved CO2 applied to animal cell culture. So my name is Olivier, and I'm going to be your presenter for the next 15 minutes. Here is the overview of the agenda. I will start by giving you some background information about the Severing-House method as the principle to measure dissolved CO2 in culture media. Then I will review quickly the most important reasons to monitor dissolved CO2 with animal cells from the bench to the manufacturing floor, the largest volumes. And finally, beyond pH control, how dissolved CO2 should be early qualified as a CPP to improve tech transfer, limit scale-down studies, or help during troubleshooting a process at scale, for example. So since almost 20 years now, the design of the dissolved CO2 probe is as displayed on the left of the slide. First engineering test of the actual design that was launched back in 2003 were conducted by 4 groups of cell culture experts from Germany and Switzerland. Some design improvements were made back in 2016 in order to improve the performance of the sensor in line for batch under perfusion, sometimes exceeding 250 days. As you can see, this is an established method used daily in cell culture the past 20 years and with patients since in hospital since the early 60s. So the method is well established and qualified for cell culture because a dissolved CO2 comparison offline and inline is using the same method, providing the same accuracy. So, and to finish quickly on the METTLER expertise on dissolved CO2 sensor, the picture on the left is representing the original design from Dr. Ingold back in 1980. But more interesting is the statement Dr. Ingold made at the time in this article speaking fermentation. The CO2 content of the gas outlet don't accurately reflect the dissolved CO2 content and its accumulation in culture media. So let's have a look at the principle and method of measurement. A carbonated buffering system inside the sensor is surrounding a micro pH sensor that is measuring variations of dissolved CO2 content in culture media. So dissolved CO2 is passing the sensor membrane to be quantified. The principle for measuring the pH is explained by the Severing-House equation as displayed. Important to note for cell culture application, this is a selective pH measurement method based on CO2 content in media. And somehow there is an analogy when looking at CO2 diffusion passing both ways a cell membrane, speaking of the principle. And regarding the robustness and performances during cell culture, I will briefly approach those aspects in a couple of slides. Let's take a look now at dissolved CO2 with a process perspective and the reason for monitoring dissolved CO2 from preclinical batches to commercial manufacturing operations. So nothing new when saying a growth of feeding media, cell culture media is a complex mixture of materials that are difficult to track in line. Thinking of depletion, for example, of an antioxidant or a vitamin or a chemical supposed to feed and maintain healthy cell population. It's hardly difficult to find a link and connect the dots where here simply because media has an oxidative index that is changing continuously, and such media voltage variations are impacting pH stability and the buffering system if any is used. So if you look even separately, thinking of dissolved CO2 and pH, dissolved CO2 fluctuation and accumulation at various rates is impacting pH control. And an existing buffering system could reduce or amplify the impact on cell population and excreting, of course, more and more dissolved CO2. So pH will definitely be essentially affected by a dissolved CO2 variation in media. It means that at fixed inputs with, speaking of temperature, pH, aeration, the accumulation rate of CO2 and its rapid variation is going to affect directly the pH stability that is required and impact the process. And this is difficult to track if you don't look at CO2 as a critical parameter affecting the pH. So that's important to be sure that at least CO2 to pH and pH to CO2, both accuracy are maintained all over the process. So for pH, it is important to always remember that a buffered system will artificially impact the pH at desired set point and ultimately impact its control since dissolved CO2 is accumulating. And it's necessary to strip this excess of CO2, which will also influence the formation of foam and ultimately affect the pH, again, its fluctuation, and therefore its control. So pH, temperature, agitation, oxygen, feeding nutrient, etc., are mostly regarded as inputs, but could become valuable to better manipulate them with outputs as a dissolved CO2. Other outputs, such as media osmolality, the accumulation of byproducts as lactate and glutamate, unwanted oxidative stress conditions that are also going to be impacted by dissolved CO2 and how to maintain it at a desired value. So maintaining a desired dissolved CO2 content in media with an increasing cell population is going to impact positively pH control at any scale. First graph, the overall concentration of carbon dioxide gas required to maintain a pH of 7.1 was high, and this negatively influenced cell population. As you can see on day 9, when the cells began to descend into the death phase, the aeration of CO2 required by the system decreased and fluctuated until the end of the culture. It was hypothesized by the author that the fluctuated and high concentration of CO2 has a negative influence on the cell growth and eventually protein production. Second graph, same experiment, but modified gas supply for the CO2, for the dissolved CO2. So the maximum viable cell density achieved during the process was above 26 million per ml on day 8, and the viable cell density was comparable to the golden patch. And the successful transfer of process in terms of viable cell concentration to the 2-liter bioreactor was achieved. So, and viability beyond 96% was a final product with a final product concentration around 2.9 grams per liter. So another major difference was the addition of feed to the bioreactor. Since the cells follow the expected growth pattern, the feeding continued as planned due to the demand of nutrients by the cells. The amount of CO2 sparged into the bioreactor and feed medium supply has a direct influence on the cell growth. Interesting finding that suggests to look at how CO2 as a gas, when sparged into media and dissolved into media and dissolving continuously into media, is affecting the process. It's also confirming at bench scale to better look at CO2 management and how, for scale-up, it will impact pH control. So to optimize pH management, it's essential to better manage CO2 accumulation in culture media. In this case, whatever the origin of the pH difference of 0.3 pH unit, from inoculation, it will impact the three major outputs of the process. The design of the pH sensor will influence the overall performances in line, including post-sterilization, the eventual adjustment required. And of course, the impact of the dissolved CO2 variations at various pH from 7 to 7.3 are not tracked here continuously to detect this pH anomaly. So coupling the pH and dissolved CO2 sensor to better characterize the impact of pH variation will help to better characterize the impact of dissolved CO2 on pH control at any scale, starting at small volumes. So today, with a dissolved CO2 sensing solution performing attitude in the bioreactor, this relationship between dissolved CO2 and measured pH in cell culture is key to develop a tightened control pH. So first comment has been made with an engineering perspective, confirming the accuracy and performance in a commercial environment for a dissolved CO2 sensor, measuring in line precisely, and as you can see, beyond 300 days for a single perfusion batch. Most of our clients working in perfusion or in fed batch up to 25,000 liters found at least important to monitor dissolved CO2 batch to batch. They see the possibility to better monitor CO2 as an input or an output more precisely. The second sentence from Mr. Ken at Lonza as well also summarized with a process science perspective the impact of dissolved CO2 upstream and downstream. Finally, and to wrap up with the process values when measuring dissolved CO2, let's quickly look at the possible application beyond pH control. So past 40 years, many publications are mentioning the benefits for tracking the dissolved CO2 content in culture media and as a stress factor that would impact downstream operations. So it's interesting beyond the impact on pH alone to look at dissolved CO2 with different perspectives, essentially a stress factor. So impacting directly productivity, batch to batch, and quality attributes. Measuring dissolved CO2 is often neglected in cell culture and fermentation, but it's a well-established technique for cell culture, and it was used even before first commercially approved monoclonal antibodies back in the early 90s. So Dr. Ingold has invented first industrial dissolved CO2 sensor for fermentation applications, describing already the challenges in large fermentation tank, describing and facing the inhomogeneity issues affecting productivity at larger scale. So dissolved CO2 is a very dynamic parameter simply because the dissolution rate of CO2 is about 25 times higher than oxygen in water at 37, and it's faster at lower temperature and higher pressure. And dissolved CO2 will impact growth rate, quality attributes of the protein, or the purity of an antibody targeted. So from the bench to very large scale, in suspension, including various single-use bioreactor design, the CO2 impact will be different. So it's important to qualify the dissolved CO2 in order to monitor and control its accumulation in media at various scales and various bioreactor design. Cells, they have to adjust continuously to their environment. Even if sometimes a bioreactor might sound or look like a black box, mapping inputs and outputs while trying to characterize the factors that influence performances at scale is an approach helping to improve performance of a defined of a process with defined process parameters. As an example, a set of inputs and input could be drafted like this when early looking at parameters that would impact performances and goals when designing a process model. And this is why beyond pH control, measuring dissolved CO2 could become more valuable as a CPP. So beyond pH control, dissolved CO2 in media will impact metabolic fluxes to be used to optimize a feeding strategy. For example, a feeding based on not only the carbon source, it will impact CO2 will impact the intracellular pH, will impact cell and media osmolality, and dissolved CO2 will impact quality attributes that could also complicate downstream operations. Even if for an existing process model, monitoring dissolved CO2 don't make sense simply because it has never impacted your process model as of today. I hope this quick presentation, has made you more curious about dissolved CO2 characterization at a various scale and culture model. Finally, as ORP variation in culture media upstream, dissolved CO2 is beyond the value of the CPP alone, but also a critical stress factor to better qualify with a quality perspective and other form of stress factor like the generation of ROS or RMS. I think it's ending my presentation for today. Thank you for your time. And someone will take the lead from here.
Are you ready for some questions, Olivier?
I am.
As a reminder to the audience, you can go ahead and type in your questions, and we'll be starting question and answer now. The first question is there a good CO2 or dissolved CO2 measurement system for the Amber 15 and 250 systems, or is the METTLER TOLEDO dissolved CO2 sensor good in any bioreactor, or has it only been tested in some?
So at the scale of the Amber bioreactor, it's already a micro scale. It's not so mini or micro scale, whatever. But the miniaturization of the technology is possible, but has not been achieved yet.
Okay. And how was the optimal dissolved CO2 level chosen? Was it a constant or a set point, or plus or minus a dead band?
But there is various approach here. One of the approach with the buffered media is a fixed point to monitor the similar relationship between CO2 and pH and lactate accumulation. It has been reported in various publications, or depending of the stress factors or the way the process has to be under stress with the different amount of CO2 accumulating. It could be a different approach where for example, if the process has been defined starting maybe at 50% accumulation, down to 44%, and get them at a higher level around 10%, this profile defined at a small scale will be used to copycat actually this profile in very large volume.
Okay, great. Well, I think we are out of time for questions. If you still have questions, go ahead and type them in to the Q&A window now, and we will pass those on to Olivier, and he can follow up with you directly. So thank you to our audience for joining us, and thank you, Olivier.
Thank
The
you.
recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcast. Look for those announcements in your inbox. Goodbye.